everybody. Welcome back. So I am Sky Long. I'm a graduate student at UMass, and I am incredibly happy to welcome you back to a new series of OEB Science Cafe. And I see a lot of new faces, so that's really exciting. We've been doing this now for about 3 years. Is that right? I'm going to make up a number. So we've been doing this for about 3 years, and we're back for another great series. Tonight we're going to be talking to Dr. Courtney Babbitt. We also have to thank all the wonderful people who support us in the community. The Organismic and Evolutionary Biology Graduate Program at UMass, the Society for the Study of Evolution, the Natural History Collections at UMass, Mass Academy, and of course, Essalon Cafe for providing us with the space, with the food, with the baristas, with the drinks. Thank you, Essalon Cafe. So I would encourage you to get a drink, tip generously. These people are here after hours for us very specially, so be sure to be gentle to your baristas. And thank you. So we will get into Courtney. All right. Hi.
Hi.
You want to turn on your microphone here so we can hear you?
Is this on? Yes.
Is it on? Everybody can hear? Great. So, Courtney, you are a professor at UMass. That's correct? Yes?
Yep. That's correct.
And we just got to make sure, right? Because you're new at UMass, so you might have just snuck in. You might, right? You try to trick us.
Yeah.
Right. Okay. And so you do some really exciting work. You do some work looking at the genes that make us basically who we are and what we are, right? Not me specifically, but us as a species. Us as a species. And some of the things that you look at has to do with diet and some of the things that we eat. And so while we enjoy this wonderful food that's provided to us, I know that food gives me energy, right? I eat it, I get energy, I do stuff with it, I grow with it. But I don't think that I really understand how that energy is dispersed in the body. So how that's used within my body.
Okay. So what makes us, when I do comparisons and want to find out what makes humans unique, I compare them to other primates. So our closest living relatives are chimpanzees. We have some skulls here so you can see relative brain size. Here's a human skull, and you can see that the volume of the brain size is much larger. And that remains true even if you scale for height and stuff like that. But what's actually going on there, it's not just a bigness. That doesn't make it, that's not what's really interesting about the human brain. It's that this is taking up 20% of the energy of your body, even at a resting state. So you're not doing anything difficult, you're not thinking hard. Just when you're resting, this is 20%, this brain is taking up 20% of your energy, whereas in non-human primates, it's something like 80%. So twice as much energy goes into serving your brain than it does in our closest relatives.
So I'm just sitting here, I'm doing absolutely nothing. 20% of what I'm eating, 20% of the Doritos, the Cheetos, all of that directly into my brain.
Exactly.
So that is insane. The brain doesn't do anything. I mean, I'm a graduate student, right? So my brain does absolutely nothing most of the time. But the brain isn't running, it isn't jumping, it isn't beating, it isn't breathing. I mean, what's going on in the brain? Why is it so stinking expensive?
So there are so many connections that are happening between neurons that it's not that chimpanzee brains are simple. They look very much like ours, except we have a much larger layer of gray matter on top and a lot more connections between and the middle. And so the energy is feeding those cells as they're connecting and talking to one another.
So the difference really between the 8% and the 20% has something to do with how connected that brain is, how connected it is with each other, and kind of how hot it's running.
Right. It's running in a very it's running faster. It's doing a lot of stuff.
All right. So we're overclocked. Right. We're overclocked, which is why we do so many weird things, and we think about it, and we write poetry, and have Justin Bieber and watch TV and things, right? Because we got 20%, we got to do something with it, right?
No, that's not.
Justin Bieber takes up like 2% at least
Of your brain.
of hating him in my brain. Yes. All right. So we have this really big brain. It's really expensive. It does a lot of things. That's got to affect us somehow. It's got to affect us physiologically. It's got to affect us behaviorally. It's got to affect something about us as humans having this giant clunking machine inside of our head. So what's that doing to us?
So that's, I think, really exciting. It's something that anthropologists have known for a long time, is that if you measure brains, we're using a lot more energy, and so there might be trade-offs. And this idea of bigger brain, but maybe our muscles are weaker, or maybe we have less of them, our guts are much shorter, and that is going to relate to what we eat. And so there are the idea of these trade-offs, that to let this run, we have to change other organ systems and give less energy to those organs.
So because we're running so much up here, we're not capable of running, because there's maybe a limited amount that a body can take in, that a body can work with.
Right.
There's some sort of limit to how much energy that can take in.
Yeah.
Okay, so we're going to pause quickly for a question. So if anybody in the audience has any questions about what we have gone over so far, just to let you know, we open questions up so you can raise your hand at any time. I may ignore you. But I will try to get to everybody. We'll try to get to everyone. We're going to take a couple breaks and then have a big question and answer at the end. So if we have any questions about diet or what we talked about so far, I see questions in the front. Yes?
What makes it take that much energy? Is there like tiny cells that just take some energy and make rock?
Okay, so is it the tiny cells, the neurons within the brain using that energy? So is it something specific about maybe a human neuron compared to a chimpanzee neuron that's causing it to use? Is there something specific about the neuron?
That's something that we don't quite know, but yeah, it seems like our brains are using energy in two different ways more than a chimpanzee does. So they're hungrier, and so they need more energy to run.
Okay, so there may be something with the cells within the brain. Yep.
Smaller? Does this mean the smaller you are, the smaller your brain is?
Bum, bum, bum. We asked from the very tiny girl over there.
Relative to size, I mean, it is smaller, right? Because you can measure this volume inside of here and get a measure of brain size, but within humans, this variation of brain size, it doesn't really have an impact, within normal human variation. So having a bigger head doesn't make you smarter.
So if I was to grow a gigantic head, say, due to radiation,
This big.
this big,
That big?
would I be
Big as this
that much
building.
smarter?
That would be outside the normal variation in humans.
So I
You might have a superpower at that
I
point.
might have a superpower.
Yes.
That's something to look into, folks. We don't have any insurance here, so if you do your own experience with radiation, do not blame us. But I'm curious to know. Okay, so larger brains, very hungry, affecting us by maybe making us have less muscle mass. Not me, obviously, I have very big muscles. So I'm thinking about the diet, I'm thinking about the things that thinking about how that must have affected us. So presumably, chimpanzees and humans, we have a common ancestor. We came from common ancestry. Presumably, earlier on, we probably ate very similar things. So what was our diet like 2 million years ago, and when did that begin to shift to something that supports a giant brain like this?
So when you look at human ancestors, older humans, their brain is much smaller. It's much closer to that of the chimpanzee. We shared a common ancestor about 6 million years ago, which is a really long time. I mean, it's hard, right? The summer went by fast for me, so it's hard for me to really understand 6 million years. But that's
You get a lot of research done.
maybe. But for the first few million years, the brain size of our ancestors stayed fairly small. And about 2.5 or 2 million years ago, there seems to be a couple lines of evidence that our diets have shifted from more plant-based and tubers, things like potatoes or yams, to a lot more meat eating and eating a lot more meat and fat. And that is correlated, and it's not necessarily a cause, but it's correlated with timing of evidence of hunting and finding fossils with much larger brain sizes. So there may be a lot of impact that our diet had that sort of played off one another.
Now, I love that you brought up the term correlation and causation, because I think that causes a lot of confusion. We're going to be talking about a lot of things today that are correlated.
Right.
So things that are correlated are things that tend to change together, but not necessarily things that are directly related to each other.
Exactly, and it's really hard for us to figure out in evolution because we can't replay the experiment. We have to just know that, you know, correlations can be informative, but not to take it too far.
Great, so that's really helpful. Yep.
You said there was evidence of hunting. Is there also evidence of social behavior?
Okay, so hunting and maybe a link between hunting and social behavior.
Yeah, there's some evidence of, you know, they'd be working together in groups and in creating stone tools. It probably progressed more like our ancestors would find a carcass that a lion had killed, and there'd be some meat on it, and okay, we'll eat this, it's easy energy. And then it became, it's called passive scavenging. And then it became a little bit more active, and then that probably then went into hunting in groups.
So, for example, kind of what is happening over here is passive scavenging, right? And if I actually have to walk to the McDonald's, that's where active scavenging.
Right, then you really need to
have to get tools and everything. It's very expensive. All right. Great. And so as this begins to shift, as these things are beginning to shift, I'm going to let you kind of have the lead on this, but there's a huge change in human history and human evolution that kind of changes everything. And so I'm going to let you kind of, what is that giant thing that's going to kind of change us and make us who we are?
Yeah, so to iterate where we've been, we're shifting into eating more meat and more fat in our diet. And then about 10,000 years ago, we settled down in communities and started to domesticate crops. And so that was this huge shift from hunter-gatherers to cities, right? And groups of people sitting in a stable place, growing crops and domesticating both plants and animals like cows.
Great, so then we kind of move into more domestication, we're domesticating things, we now have animals, right? We don't have to go chase a cow down, cows sitting in the backyard, right? This makes things so much easier, it's much happier. And this is obviously probably having huge effects on our society. And so causally these things are related with an increase in brain capacity or at 10,000 years? No, we already have the brains.
We already have the brains. It's just a shift in what we're choosing to eat and where we're choosing to get our energy from.
Okay. So a huge change like this is probably going to be affecting other parts of our brain. And so when I, as an evolutionary biologist, I always think, gosh, this has got to be affecting our genes in some really shocking, huge way. So have we seen any effects of this in our gene code? I mean, have we evolved within the past 10,000 years beyond just evolving away from chimps?
Yes, so one of my favorite examples is about lactose intolerance. Is anybody lactose intolerant that they want to raise their hand? A couple people, maybe I get some nods.
I'm very sorry for you. Cheese is the nectar of the gods.
I'm not.
Is anybody lactose tolerant? Can you digest milk proteins or cheeses or whatnot? Okay, so those of us who have our hands raised are actually the mutants in human populations, myself included.
That was not a question.
Most of humanity can't digest milk after childhood, right? Most people on the earth cannot do that. This idea of lactose intolerance is really more of a marketing thing to people from European populations, right? Lactose intolerance isn't a sickness. I mean, I do feel bad for you because cheese is really great, but like, it's not a sickness, it's a totally normal thing. All mammals are able to digest milk when they're babies, and then they go through a period of weaning, and then they don't drink milking. It's strange that we drink the milk of.
It is a little odd if you think about it. Don't think about it right now if you're eating, but if you think about it, it is odd that we continue to eat the breast milk of another animal. Let's admit what it is. And it's fermented delicious products. Well after adulthood, right? So that's a really strange kind of shift in diet. And yeah, well, I was going to ask if anybody had any questions about so far. I see great hand raised. Yes.
When did they start cooking?
Oh, when did they start cooking? So cooking is something that's really interesting. What does cooking do, and when did it start within humans?
So the when did it start's a little controversial, but at least 400,000 years ago. Wow. And maybe more than that. But maybe farther back than that. And cooking breaks down, especially meats, and makes it a lot easier for us to digest.
And so that may be related to the fact, so you talked about that we have a shorter gut, And so having that shorter gut makes it more difficult for us to process certain types of food, is that correct?
Right, and so cooking essentially gets the nutrients to us more easily, and so we maybe we're able to shorten our gut and not spend the energy processing nutrient-poor foods that are really fibery, husky kind of foods.
Okay. So we can do the important.
So we can do the thinking about it.
Which is very important. All right, any other questions? Yes?
So you're saying that lactose tolerance is the mutation. So how did that come about? People just had the cows and started to.
Yeah, so the question is, lactose intolerant or lactose tolerance is a mutation. We're the weirdos. And so how did that come about? How did that kind of come into the population?
So the first way it was studied is that it's really prevalent in people of European descent, from certain European populations. And it's really cool because you can actually, if you take a map of Europe from north to south, the Scandinavian countries, it's almost at 90 or 100% that mutation, everybody's got it. And what that means, and it's the strongest signal of natural selection, positive natural selection, that was good for these people in their genome. And so what that means is the people 10,000 years ago, 8,000 years ago, who started drinking this milk, they were able to have descendants that lived, they had descendants that lived till today, whereas people who didn't have this mutation didn't probably pass on that gene, or this mutation, to subsequent generations. So it's really, really helpful.
So there was something about it within that population, within that northern population, where we see that increase survival rate in people who are able to consume. And that might be because there's something about milk, there's something about those products that's very beneficial to us.
Yeah, so what's neat is you can actually go from north to south in Europe and see that it's like everybody's got it in Scandinavia, but not so much in Italy, kind of half the population has it. But what we think is that milk has a lot of vitamin D in it, and these people are in a very, very dark environment, much of the year, they don't get a lot of sunlight, and so that would convert an advantage to them if they could get vitamin D through drinking milk. And so that's one of the things. But what's really cool is that this mutation has actually evolved in every group of people that aren't related to each other necessarily across the globe. So there's another group in Africa that they've domesticated cattle. They've sort of in Kenya, Tanzania. And the mutation is 4 base pairs away from the European mutation, but it does the same thing. It confers the same, you're able to drink milk into adulthood, but there it's possible that it's not vitamin D, that it's more an access to clean liquids, to clean water through the milk. That you don't know if the stream is good, but you know anything coming out of the cow is probably very healthy for your children.
Oh, so that's really interesting. So here you have a single source domestication of livestock, which provides this product. And in one area, because the environment is such that there's a lack of vitamin D, it confers this vitamin, the ability to drink it confers some benefit to them to having that vitamin D, but in a totally different environment where they're getting tons of vitamin D, this is conferring a totally separate advantage, maybe from drinking the liquids, but those mutations are incredibly close to each other.
Right, so the human, that's the wrong way to say it, but the genome's figured out the same solution 2 different times.
So I do want to quickly, is there any other questions? Because we're about to move on to, yes?
Is that small difference in code something that has to do with the activation of other bigger pieces of code that are already there?
Oh, see, you're jumping ahead. You're jumping ahead, my friend. Sneaky. I bet you're one of those lactose tolerant people, aren't you? Yep, I gotcha. So we are gonna, that's a great spoiler into the next thing. So I'm gonna have you guys hold your questions because we're about to get into some pretty deep stuff. So one of the things that I think is really important, and also a source of confusion, is the word gene. So gene is something that I think we've all heard, we've all read, we have kind of an understanding in our head about it. But I found even my understanding of what a gene is different from what my understanding of a gene was when I was an undergraduate, which was 1 billion years ago, right? I'm very old, ancient, don't laugh, it's not funny. It's true, I'm ancient. And so our ideas of what a gene is has evolved very much. So can you kind of give us a sense of what our understanding of what a gene is now?
So a gene is effectively a stretch of DNA that it's a recipe, and it's going to tell your cell what to make. So it's a recipe for what to make, and so it's going to make. It tells the cell how to make a protein, and the protein is something that actually does something in the cell. So it could be part of a fiber that makes your heart beat, or it could be part of the keratin in your hair, and that actually does something. But the genes themselves don't do anything. They're the instruction to then make something that's helpful for the cell.
Okay. And so, yeah.
Are genes only parts of the DNA that actually code for specific proteins, or can they be bits of DNA that turn the genes on and off each other?
This goes to the next part. So this is great. See, we're leading you along the path. So the question is, are genes just things that code one thing, or can genes affect other genes kind of more directly? And so one of the things that we should talk about is the idea of what a gene product actually is, and what a gene produces, and what those things are capable of doing.
So yeah, one gene can produce something very simple like one gene, one protein. But most genes are much more complicated than that, and they've got sort of bits you can switch in and out. They're very modular, so that you can make lots of different kinds of proteins depending on if you're a brain cell or a heart cell or a skin cell. So you can have all the same genome in all of your cells, but make very different proteins from those genes.
So that's a really interesting thought. I don't know. This is one of the thoughts that I had, admittedly, back in my day. This is when I was an undergrad. The thing that blew my mind was that every single cell in your body has the exact same genome.
Thank you.
Your eye cell has the exact same DNA that your stomach cell has, that your liver cell has, that your skin cell has. All of those are exactly the same, but there's a point at which they differentiate. And it's not that they have different genes, it's that those genes are acting in a different way. So some recipes are turned on, and some recipes are turned off.
Exactly.
And to further complicate it, right, because we like to complicate things in science because it makes us look very smart. Right? It's okay. It's very
I'm smart too.
Yeah. So genes can actually affect other genes.
Exactly. And so the human genome has about 22,000 genes, and it'd be easy to understand if one gene made one protein and just did it in one tissue, right? Just working your heart.
Sure.
Your heart gene. But it's not
And you
like
only have to understand 22,000 things.
Right.
Smart about that.
but we have all the same genome in every one of our cells, and it's just being turned on and off to make these different kinds of cells. But all of these genes, or most all of these genes, are interacting with each other. And importantly, there are a lot of genes that their only job is to turn on other genes in specific tissues or times during our lifespan.
So therefore, evolution can act not only on a single product, right? It can not only change a gene that produces just a protein, it can affect a gene that affects hundreds of other genes.
Right, or it could do that just in one tissue and not another.
Oh, tricky.
Right, so it could have evolved a real change that happened in brain tissue that activates lots and lots of genes, but that doesn't mean that it had to happen in any of your other tissue.
And so it's working just in the brain tissue, but say in the heart tissue or in the liver tissue, it's just doing the normal thing.
Yeah.
That's amazing. So that really gives evolution, we could say, a really broad palette with which it can work with, because it can affect multiple things by making very small changes.
And that's why even over what we think of as short evolutionary time is 600 million years, you can have these really, really big changes, even though our genomes are 99% similar to chimpanzees.
Yeah, which is shocking, that we have 98% similar genomes, but we're able to do so much with it. That's cool. That's very cool. Yes?
I'm backing up a little bit, but this is kind of making me realize that my idea of natural selection is very kind of brutal sort of image in my head. You know, so I have this image when you talk about people who are being selected, you know, they're being favored by natural selection to have to let this mutation develop. You know, this lactose tolerance is being allowed to develop. I'm getting this image of a world in which there's nothing to eat but milk.
Okay, so I like this. This is great because I think this gets to a central, kind of maybe a central misconception that a lot of people have, and even a lot of scientists have. So let's talk about, I think this is perfect. Let's talk about natural selection. Let's talk about how that works. And let's talk about it from the story of lactase and the gene of lactase. So we have a group of people, they have regular genes, they're lactose intolerant. How do we go from a population of people who are lactose intolerant to a population of people who can eat nachos like nobody's business?
So,
Okay, so this is the world of milk nachos.
Yes.
yes. It is really more that they may have had many other things to eat, but it's that slight advantage that the people who could drink milk, if that mutation arose in the population, and then that family realized that, oh, we can all give the kids milk, and oh, our kids are, you know, bigger and stronger than the other kids. That slight advantage, over lots and lots of time, you'll see that one gene spread where their kids are big and strong, so you want to marry their kids, and then they have more kids, and that's how it spreads across the population. So it doesn't need to be a big thing where it's like milk or death. but then even that slight advantage can then will over time take over in the population if that advantage keeps being an advantage. Right. And now, you know, you can take vitamin D supplements, it's not as much of an advantage, but during those, 10,000 years ago.
It's a delicious advantage.
It is.
So I think that's really interesting because that's the idea that we have of evolution as an all or nothing thing. So evolution works on a population, right? Natural selection works on an individual. And so evolution is really, in a really technical way, it's the change of allelic or gene frequencies over time, right? So it's how frequent a gene is in a population over time, is that right? Mm-hmm. This is going back to my bio 100 days, so bear with me, I haven't done this in a long time. I've been in a hole, so here we go. And natural selection works on all the genes in your body at one time, right? So everything in your body is being acted upon at one time. And things don't have to confer a huge amount of selective advantage to make that gene increase within the population. Mm-hmm. So you said that lactose intolerance is a really strong selective signal. And so maybe you can describe what a strong selective signal means. What we mean when we say that has in the pop. I see you smiling like I just asked you. Can you derive that equation?
Well, what we can see is so you can see the mutation, and then you see around it, in these populations, there's not much change. And so it's essentially, a way to say it is that it's stable. It's stable. It's gone into more people in the population very quickly.
Yes.
And so it's a very good thing. Evolution's not still playing around with it.
Because if a gene, so let's say if a gene isn't being used, right? That gene can have mutations occur in it that makes that gene not work anymore, and natural selection doesn't care.
Right. Because sort of mutations just sort of accumulate over time naturally. It's not a bad thing necessarily, but if it's in a gene, doesn't matter. No. If it's in a gene that's really important, bad.
Yeah. Obviously bad.
But here it happened to be something that conferred just a slight advantage to people, and maybe they could get a little more vitamin D or clean water or whatever it is.
And those mutations that occur in the genome, they can be mutations that don't mean anything until magically something within the environment. So it's possible that, let's play what if, that this lactose gene maybe mutated pretty early on, but it didn't really mean much until that population went to an area where suddenly the ability to drink milk conferred a slight advantage.
Right.
And so they produced more offspring, they produced more children that then had that gene. Mm-hmm. And then that gene suddenly, natural selection goes, that gene's important, and watches it very closely.
Exactly. So what's cool about it is we all have the gene to break down milk, to break down milk sugars, lactase. So what happens with most of us, or with most of humans, is that gene's expressed at a kind of low level while you're nursing, and then it gets shut off during weaning. And what's cool is that this mutation is in a gene. It's in a region that's like, it's really, really far away, 14,000 base pairs away, that then loops over and brings other proteins with it, and then that elevates the expression of the lactase genes so that you're still able to break down milk sugars.
So in other words, that gene's not producing a protein. That mutation's not producing a protein.
Exactly.
That piece of DNA is actively acting on that gene.
So that one base pair will bring some proteins with it and loop around in this really cool, like, I mean, think about it, like this big stretch of DNA is now touching another stretch of DNA. There's a big loop here, and now that gene can be expressed, and now you can break down milk sugar.
That's great.
That's crazy. It's the gene producing that's activating the other genes.
Great. So how is that gene actually activating that gene? How is that actually working? It's not producing a protein. It's somehow magically making this happen. How's that working?
So it's actually one base pair mutation that will bring down something called transcription factors,
Transcription factors.
which are just proteins that turn on other genes. It's not a big deal. But that one base pair will bring transcription factors that are really important, and then that sort of cascades and other machinery will come down that will then express that gene and make the protein that can digest milk sugar.
Yeah, so genes themselves obviously produce proteins, but in this case, this gene is just taking proteins that's attached to it and bringing it over.
Yeah, it's not even, it's just that one base pair mutation. It's actually in the middle of another gene. It's really weird. It's in the middle of another gene. But it's enough to sort of to attract it. You can think of it as just sort of like a little sticky paste that then will loop over and allow for gene expression.
That's amazing. Yeah.
I have two questions.
Okay.
One is, you said that some genes, you've been talking for a bit of some genes to another genes off on.
Okay.
Now, I would imagine that there would have to be some genes, if genes are the only thing, like, into the author genes, DNA, or maybe. But I would think that something would have to turn on both, like say, okay, I'm turning on these two genes, those genes, and then turn it off. Everything starts off, and nothing can be on until something else on. And my second
Well, let's start with the first one because that is a huge question. So you asked the chicken or the egg question, right? Which, by the way, it's an egg. Right? Because dinosaurs laid eggs. Okay, so the question is, we have genes that act on other genes, right? What's the gene that tells that gene to act on another gene? So how does the whole thing get started? When you're two people love each other very much, like your ears, and a stork comes. What's happening there? How do these genes get started? You have sex cells that have genes in them. Those genes aren't really doing anything. They're sex cells. They come together, and then something starts.
Yeah, so the way that it works is that, does this turn off? So the egg, I mean any kind of egg, so you can think of a chicken egg, when that gets fertilized, it has a lot of instructions to go, okay, well, I have all the instructions to get started. So I'm going to tell you what to do before you start making your own proteins from the...
And that comes from mom? So
that just comes from mom.
Okay.
And then as the cells start dividing and you get, a more complicated embryo, then the embryo sort of turns on its own genome. It's like, I've got this, thanks. So
Kids. Are those genes real jerks? I'm kidding. Kids are wonderful. I don't have any. Okay, so you have mom's genes are saying, here's the blueprint, here's how to start. And then those genes, as those genes start to go, cells then, due to a very complicated kind of evo-devo reason, but then they begin to shut down things and what we call differentiate into specific cells. And those specific cells have a book of recipes that say, I'm a liver cell. And so this is the book of liver cell recipes that I have. And I will only use these liver cell recipes.
Right, I've got all the other recipes with me, but I'm just going to put them in a corner and not worry about it.
And I'm not going to worry about it.
Yeah.
Does that make sense? Okay, so is it a one-way street or can we ever talk back?
So there's a lot of talking back.
Yeah,
It's messy.
just like kids. A lot of talking back.
It usually starts with one or two master genes that will turn on like hundreds of genes, but that doesn't mean that those hundreds of genes can't then turn the master ones off. That happens a lot. So there's a lot of what we call crosstalk between the genes, and that changes as as over time, it's different in a little embryo than it is in your mature liver cells.
So that would probably be why it's so difficult for us to track down genetic diseases and genetic kind of changes within the body, because it's not a one-to-one relationship. There's no gene that says, I'm a cancer gene.
No, every gene that we have, the ones that are functional, 22,000 of them are meant to be there. It's just when one goes awry, there's something that changes in it. That's going to affect hundreds, if not thousands, of other genes that are going to affect thousands of other genes, and that's going to be different in different tissues, across in different people. So that's why we call them complex diseases, things like cancer or diabetes, because they're really complicated. It's all these different things are interacting with each other, and it's a really dynamic world in there. Really.
And that's why it's really difficult, I think, for us to read in the newspaper, when they, everybody wants to cure cancer, but cancer is not one thing.
Right.
It's a complex and difficult thing. And so that's why we should support people who do genetic research.
Yes.
Yeah. Give me money. Yes, another question.
So we're talking about genetic diversity within cells. So if we're talking about lactase, what cells are actually using that gene?
Oh, good question. So what cells in the body? So we know that cells are different within the body. Cells have the same genome, but they use different genes. What cells within the body use that lactose gene that's actually doing the work?
The cells in the lining of your small intestine are actually, that's the very specific location where you'd be able to break down milk sugars. And I think I've heard tales that if you digest a lot of milk sugar and that gene isn't activated, it's very uncomfortable. I'll leave it at that.
We'll all leave it at that, yes. So, all right, so it's in that lower intestine, so it's in that gastrointestinal area. That's interesting. But that mutation is still present in all the other cells in the body.
Exactly. So you can figure out, we call it genotyping. You're just actually finding out if that base pair is one thing or the other, using a piece of your skin or a little bit of swab from the inside of your mouth.
So you don't have to actually remove a part of my small intestine,
Yeah.
which I appreciate. Thank you.
What does it physically mean for a gene to be expressed? So you have the order of base pairs, you have the genotype, and we all kind of know what that means in terms of the 4 bits. But what does it mean, physically or chemically for a gene to be expressed or not?
Okay, guys. So we started out. So does everybody know what DNA is made out of? So everybody knows that, right? And so what you're asking is, you have the DNA, we know what the genome is. What does it mean when the difference between a gene that's just sitting there and a gene that's actively being expressed? What do we mean when we say that?
Yeah. And can you look at the string?
Oh, can you look at the DNA and say, okay, so is there a change in the DNA itself that says I'm being expressed, or is there something else we have to look at?
Secondary effects that you're looking at.
Secondary effects. So how do we tell that a gene's being expressed? What do we look for?
Yes, so what you can see, so DNA is a long string. And like any long string, you imagine a ball of yarn that if you wanted to keep it really organized, right? You don't want to let it sort of stop around. So what we can see is that most of the genome is really, really organized and wrapped around little balls. It doesn't matter what they are, but it's very tightly wound. And if a gene's being expressed, what you'll see is that the area where the gene is actually unwound.
Oh, interesting.
So the gene is accessible to the rest of the cell to then copy it and then make a protein from that. So you can, the chromosome itself is open in places where the gene's going to be transcribed and translated, or the 2 chemical steps of it, but then essentially there are 2 steps that then make a protein from that
So
gene.
do we know how genes make proteins? Everybody know that? Do you want to tell us how? Okay. So you have a gene. This is like a test. This is like my oral exams that I had to take to be a doctor, almost doctor. Okay. So here we go. We're going to see. You can judge me.
Do you want me to do it?
Don't tell my advisor. So we have DNA, and we have a series of genes, and these are base pairs, right? So this is the G, C, T that you typically think of. And it on its own can't do anything. Well, obviously sometimes it can, but for the most part it doesn't. What it needs is another protein to come in and read that information. And so it reads that information and produces a string of what's called RNA. And it has to do that because the DNA needs to be protected. Your DNA is really important. And so your DNA is all wrapped up and it's stuck inside of the nucleus and it can't go out, it can't go and do anything, it's an invalid. And so it is protected. And so it's being read by this machine, which is called, you guys can probably hear me, is that right?
Yes.
Yeah.
Okay, so it's being read and this is called transcription. So it's being transcribed. A little piece is coming out of whatever needs to be made. And then that goes outside the nucleus into the cell. And out in the cell are these other complexes which do what's called translation. Translation takes those combinations of letters in triplets. And each order of those letters makes an amino acid. And so I have a long string, a sentence if you will, and this translation comes along and says, okay, I read these three means that I need to put an amino acid methionine on there. I go to the next one, oh, I need to put thiamine on there. Tyrosine, yeah, that's a good one. And so it keeps going along and it reads these and it puts them together, and these amino acids have positive and negative charges on them and have something chemically that makes them move around each other in a very specific way, right? In elbows and things, it's very fancy. And that produces a protein. And so it's actually a pretty complex step, and each step can be modified by evolution. So each step can be modified. We can modify how DNA, what parts of the DNA get read by putting a transcription factor on it, a transcription factor that says, read me, read me, read me. Or transcription factor that says, read me, read me. Or transcription factor says, don't ever read me. And then that piece of RNA, when it goes out, that piece of RNA can say, read me 100 million times. Or that piece of RNA can say, read me 10 times. And then RNA, that same RNA can be cut up and put back together in different ways and produced in all different ways. And when it goes out into the world, right, that RNA goes out into the world to be made into a beautiful protein, other things can happen to it. It can be degraded. It can be shut down. Not degraded in like a personal way, no one's like yelling at it and calling it names, but it can be broken down. And so there's a DNA and evolution, and this process is not an on or off switch. It's a series of fine-tuning knobs, each knob being directed from baby genes millions of base pairs away. And these knobs can all be affected by evolution in very, very delicate ways that make you exactly the beautiful, wonderful creature that you are. Right? And so it's actually incredibly complex. And there are things that affect these from your mom, your dad, what your grandfather ate, right? Maybe even. Your granddad starved, that can change the way the genes get produced. And so there's really no simple answer. Which is what's so exciting and so beautiful and so wonderful about it, is that we're only just now starting to understand the things that make us human, which is super cool and super exciting. Yeah.
My question is, if a mutation occurs within the DNA of one cell, and there's trillions of cells in the human body, how does that
I love this.
eventually, do the cells somehow communicate the error amongst themselves?
Okay.
And by the time it makes a significant difference in trillions of cells, I gotta think it's a long, long time.
So this is a great question. Yeah, I love this. So the question is, if there's an error in your cell, how does that cell get passed on? So let's say that I have an error that occurs here. How's that working? How does that work? So how do they get passed on? How do these errors get passed on?
So the errors need to occur in an egg or a sperm cell. They have to occur in, and that way they will get passed on, and then they'll be in every cell of the offspring.
Okay, all right. So you got to go all the way back to the prime instructions.
Yes, exactly.
Thank you.
Yeah, and that's actually really interesting because a lot of people don't think about that. That needs to be what we call a germline in the cell, in the sex cells. However, things that happen to the body can still affect those germline cells. And I think when I was an undergrad, we were taught that the germline cells were sacrosanct. That the only times that things happened to these things were when they were, there was meiosis or mitosis that was happening. That's when things happened. But now we know that there are things that can actually affect those germline cells from when you're living. The things that you do now can actually affect those germline cells.
Right, and so what's really cool, what's so exciting about this time is that we're starting to be able to sequence genomes and lots of them. And what we can also do is sequence, we can understand how the environment's interplaying with the genome and interplaying with germ cells too, so they can be passed on. So, and this is called epigenetics. And it has to do with when we pack the DNA, sometimes we can put little marks on it, and that can make genes more or less easy to be accessed. And so that can change, and the environment can actually change that over time. And probably the best, or I think one of the more interesting examples is from the Dutch hunger winter. It was a time when the Nazis were surrounded, I forget what city it was, but and people inside that starved, and they starved all winter, and a lot of women at that time were pregnant. And what's special about this case, that happens all over, right? But the Dutch were very, very meticulous about keeping health records afterwards. And what they found is that the offspring of those women that became pregnant after or during had a lot more susceptibility to schizophrenia and other issues that seemed to be because they were starving kind of in utero. So you can actually change the genome kind of through generations, and then that can get passed on even farther another generation.
That's amazing. That's incredible. So we are coming to a close, so we're just going to take a couple more questions. Yep.
What percentage of our code is activated?
It's genes.
Oh, what percentage? So what percentage of that DNA code is activated? Well, that's a complex question, I think.
It is. So about 2% of our genome is actually genes. The rest of it is stuff that turns genes on and off. A lot of it's junk. Some of that junk may do stuff, some of it doesn't. Some of it's structural stuff, just, there's sort of a middle part of the chromosome that just to hold it together. But it really depends on the tissue what's actually active. So it's fun, you can actually do expression, you can actually look at gene expression across different tissues now. You can see every gene that's being expressed with some of these new technologies.
That's amazing.
It's so awesome. And in skin cells, there are about 11,000 genes are being expressed, but in a given brain cell, you can get, it's usually between 15 and 17,000 genes are being expressed.
Wow. Really?
Yeah, so it can be really different between different tissues.
That is very good, even within the same individual.
Yeah, exactly.
So, okay, one question from the crew here, and then we'll get you in the back there. Yes?
How big is a cell and how small?
How big or how small is a cell? Well, it's bigger than an atom and smaller than a bread box.
Yes. It depends on the cell. Different cell types are different lengths.
And also make sure Azalea gets her questions in.
Okay, and we have to make sure Azalea gets her question?
Azalea.
Okay, yes.
And also, how many other different big changes were there, or other than the lactose tolerance and the cooking and...
Oh boy, wow. Okay, so how many changes? So this is a good question for the ages. How many changes have occurred within the human genome since we were single-cell organisms in the primordial ooze? Courtney Babbitt?
A lot of them.
A lot of good answer. That's actually one of the best answers. Yeah, I don't think that we know.
That's what's fun about it, because we don't know the answer to that.
We don't know.
So it sounds like what you're saying is there's more structure than the double helix that you referred to, and it's part of what is going to induce expression from is how the overarching structure, the details, what's going on, how is it twisted up.
Yeah.
So so does that relate to epigenetic data? Does that have to do with how it's twisted or how it's,
Okay, so the general question has to do with, is there more structure within the DNA rather than just the double helix? And does that affect expression? And how does epigenetics affect that structure, that secondary structure?
So yeah, the human genome is, I think it's 6 feet long. And you have it. Yeah, if you end to end, it's 6 feet long, and you have it in every one of your cells,
Holy cow.
except your red blood cells.
I'm super skinny then, if that's the case.
So there's a lot there. So we have to have it really, really compact for us not to be, whatever, giant. And so, yes, so there's a double helix that everybody maybe has heard of. It's sort of a famous term. But beyond that, that string gets spooled into very, very tight coils, and those coils are even more tightly compacted. And so a lot of the genome is sort of shut down for a lot of the time with only the genes that need to be expressed in a given tissue open and uncurled. And so, some of those epigenetic marks that can come from what you eat, or if you're not eating, or if you're going through a lot of stress, those marks can leave marks in the genome that can kind of kink it in a way. Some things are open, or some things are harder to open, or are closed in specific cells, and that's going to be different between different people, depending on how you've interacted with the environment and how your parents have interacted with the environment.
Wow, that's really interesting. Yeah.
So, one thing that I'm not quite clear on is how exactly do individual genes, individual cells know how to express different genes? So that, like, they're obviously using the same set of genes, so some of them are using the exact same gene to do different things, maybe, depending on what cell they are.
Okay, so how does my liver cell not produce eyeballs? So, we have all these cells, they all have the same thing. Why am I not sprouting? All kinds of weird stuff. How does a cell, a cell's not smart.
Right.
How does a cell know?
So the instructions really come from when an organism is developing. So at one point, a cell or a bunch of cells made a decision of I'm going to be allocated as liver, but I'm going to be separated from things that are allocated as neurons. And that from that master sort of, we call them master regulators. That there's essentially genes that say, okay, everybody from here on out is going to be liver-ish, right? Parts of the liver, parts of the, and that stamp of liver-ishness then is on all of those cells and all of their descendants. So that's how you get a cohesive organ from just that first cell.
So if we imagine that as a cell's developing, it's becoming more and more cells. As it's becoming more and more cells, there's kind of team leaders. And those team leaders are saying, okay, me and all my compatriots, you are liver cells, darn it. And that's all you're going to be. And then the next kind of team goes, hey, team, we're kidneys. Yeah, go kidneys. And so within that, we're then going to see shifts in structure, in transcription factors, in the production that occurs. And remember that cell then is able to pass those instructions directly on through dividing and then becoming another cell. And so it really only has to happen within a few of these master cells because its descendants all remember, oh, I only have the cookbook for this, and that's all I can do. And it's also getting information from its neighbors. So the things that are around it are producing chemicals that say, hey, we're all liver cells, right? And so it can kind of sense where it is in the body. This has blew my mind. Cells know where they are in the body by this gradient of chemicals that are around it. And so it kind of knows, oh, okay, I'm kind of here within the body and I'm doing this as I develop because of what's around me, which is awesome. Yeah. So we're going to have one more question and then we're going to thank Dr. Babbitt for going. Yes, one more question.
I'm actually working this back, but how do the organs talk to the other or stuff to tell them the instructions?
So how do organs talk to each other?
Yeah.
Do they all have cell phones? Maybe there's a body Facebook that they all go on. Is that really old school Facebook? Nobody uses Facebook. Because I'm old. Okay, so how do organs talk to each other?
They'll use the, hormones and other chemicals that they excrete. They can signal to one another.
So they are kind of talking to each other via telephone, basically. Like a really chemical telephone, a gross telephone.
Yes.
A really gross telephone. Okay, one last one.
And this is going back to stuff you were talking about at the beginning of the talk, but I was curious what your opinion is of the current paleo diet fad.
All right, so now we have some diet news. So what is the paleo diet? Should I be doing it? Will it make me beautiful? Courtney Babbitt, what do you think?
This is bad. I think that it could be really interesting. Certainly there's a difference.
So she's a scientist. She just wants you to try it and then tell her if it works. This is what you're saying.
What's terrible is I've started doing CrossFit and they're all into it, and so I'm like, oh, I've been eating a lot more meat.
You're killing my vegetarian heart right now. You're killing me.
There's a big difference between leaving out carbohydrates and what those carbohydrates are, right? So a piece of whole wheat bread that you baked in your oven isn't the same as eating a box of Apple Jacks, right? I mean, that's that quality of scale, and we shouldn't, as a society, be drinking a lot of pop and eating boxes of Apple Jacks. That's really, really bad.
Are you sure?
We've not evolved to consume those kind of
I'd like you to rethink that.
Your all Cheetos diet.
I'm my all Cheetos. I'd like to talk about the all Cheetos diet, which is a diet I've been developing. See how well it's been working.
But I think there is evidence. There was an article in the New York Times earlier this week that showed that low carbohydrate diets seem to confer a little bit more in terms of health. But
I think it's really interesting that I think what's always fascinating to me is that we learn a tiny amount, this much, and then we confer this much. Right? There's that. So we learn this much and we say, oh, aha, we ate meat. And then we go, boom, meat must be the most magical thing on the face of the planet, you know? And so maybe we don't always think, you know, what people do and what we do isn't always the best for us, right? And so we don't really understand how that works within the general kind of thing. And I think it's hard for us because, you know, even me, as a graduate student, as I go, the more I get into it, the more I realize I know nothing about this. And it's exciting and frightening and terrifying. But it's, you know, we learn so much every day. I mean, epigenetics was something that came in when I was in grad school.
right, and all of that's new, and how we're interacting with the environment. And I think what's really cool is now we know that it's not just us, it's all our microbes that are living inside our gut are now interacting with the food that we're choosing to eat, and they can affect our behavior.
Yeah.
So I mean, it's
amazing.
kind of crazy and exciting, and that's what makes it fun to look at.
So I guess kind of the final thought would be that we never stop evolving.
Right.
We never stop changing. We are not the pinnacle of magical evolution.
You are.
I am. I was leading you to that. But not everybody else can be.
No.
Because that would be too magical.
Right.
And that, you know, as the environment changes, as the things around us change, we're going to continue to evolve, and we're going to continue to understand these things that make us us.
And we're going to continue to evolve, and our diets and our interactions with the environment are going to continue to evolve.
That's incredible. I'm so excited. It's such a great time to be a scientist. It's such a great time to be doing this, because it seems like we now have the technology to try to unravel these kind of keys and mysteries of life. I mean, and every single one of your cell is the history book of life. And that's super exciting, and that's so cool. And thank you so much for coming and sharing that with us.
Listening to me.
Thank you guys for having us. Courtney, are you going to be here for a little bit?
Yeah.
And great, so you can come up and if you have a couple other questions. I'd like to thank Eslan, of course, for not doing it. You guys. You guys are amazing. I'd like to thank all of our sponsors. Please go to oebsciencecafe.org. We're going to be implementing some new and exciting changes to the website. So if I tell you, then I actually have to do it. So, right, it's obligation. So please continue to check there, and we love having you guys here. So go forward and do science.
Thank you.
Thank you.